Hello everyone, welcome to this webinar as part of our ongoing solutions for pharma series. I am Mridul Mandal, Market Manager for Pharma and Biopharma at Himadri Scientific Instruments. I will be the speaker today and our topic is oligonucleotide analysis by LC and LCMS. In this webinar, we will be discussing Himadri's latest innovation including software for oligonucleotide and its impurity analysis. Before we start, I want to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. In this screen, you will see the following items. The slides will appear on the top middle. Directly under the slides, you will see a resource list with clickable links relevant to the material being presented today. On the top left side is the widget for questions and answers. Please submit your questions during the presentation through this widget and we will answer them to you directly after this webinar. On top of the right hand side, you can see the speaker's bio. You may expand the items here to learn more about me. On the left are survey questions that you may fill out any time during or after the presentation. Finally, at the bottom pane are the icons to bring up all these widgets in case they are minimized or hidden. All right, without further ado, let's get started. Again, if you are just joining us, I am Mridul, the speaker, and our topic is oligonucleotide analysis by LC and LCMS. Here is the content for today's webinar. First, I'll give a little bit of introduction, and then I'll be talking about oligonucleotide analysis by LC, and then oligonucleotide analysis by LCMS QTOP, and then oligonucleotide analysis by LCMS triple quad, and finally I'll summarize what are the take-home messages from the webinar today. As all of you know, nucleic acid drugs offer a new avenue for treating a wide range of illnesses by targeting disease causes at the genetic level. However, when scientists do the chemical synthesis of nucleic acid drugs that can result in impurities, which require proper separation and purification techniques. In this study, an analytical method for separating oligonucleotides of different lengths using ion exchange chromatography was developed. Further, to achieve optimal analytical performance, an inert UHPLC system, QTOP and triple quad mass spectrometers with an ion pairing buffer system were also used. These studies successfully confirmed the molecular weight and achieved sensitive quantification of oligonucleotides, showing promise for safe and effective nucleic acid drug development. Here I am going to show you our latest ELSE system and its application for oligonucleotide and its impurity analysis. Here you can see the biocompatible versus bioinert. Biocompatible system is normally compatible for biomolecular analysis, but it is still used metal in their flow paths. Therefore, it could compromise for some molecular analysis due to absorptions or other metal affinity to that molecules. On the other hand, bioinert has a peak coated like stainless steel reinforced. That means the biomolecule never touch the metal directly. Therefore, it doesn't have any absorptions or compromise for the sensitivity. Here you can see in the Venn diagram that yellow part is the bi-inert, which has the capability both biocompatible and USPLC and complete inertness. For Shimmerjo's proprietary name is Nexera XS Inert, and you can see the system on the right side. Please take a moment to look at this complex diagram without getting confused. I just would like to show that all the sample flow paths of the instruments are metal-free, especially peak lining stainless steel, which means the peak tubes are reinforced with stainless steel that assure both pressure sustainability and inertness. You can identify this as the red colored lines. The blue colored lines represent titanium hydrogen alloy, which is used for solvent delivery that assures corrosion resistance. You can see on the bottom left side, the high pressure valve starter consists of alumina and DLC. DLC is diamond-like carbon, and the valve housing is made of non-weighted salts. As the detectors on the right side on the top, we have three types like UV, PDA, and fluorescence. The cell types of the detectors are as follows, like number 1, USPLC inert cell, and number 2, low diffusion inert cell. Both of the cell housing and piping are peak coated. All these things together made this instrument really useful for oligonucleotide analysis without any compromise. Here I am going to show you some results on oligonucleotide analysis by LC. Here I am going to show analytical performance in nucleotide analysis. Upper table show the type, system, and column used for the analysis. As you can see, we used both standard USPLC, Nexera XS, and metal-free sample flow path USPLC, which is Nexera XS Inert, and we also used conventional column and metal-free columns. Sample solutions were a mix of AMP, ADP, and ATP with 50 microgram per ml. Left-hand side shows the chromatogram, which is used by conventional USPLC system that has tailing. You can see first two peaks. That's why it didn't get separate well. On the other hand, the right-hand side is the chromatogram obtained by Nexera XS Inert. There is no tailing. Peaks are separated with high resolution. So the metal-free flow path helps obtain better separation and resolutions. Therefore, it is very much useful to do the analysis of oligonucleotide and its impurity. Here I would like to show you symmetry factor and area for the analysis of ATP by the number of injections. So left-hand side, you can see is the symmetry factor and the number of injections. For standard USPLC, it's orange color, and for Nexera XS Inert or bi-inert is the blue color. You see the symmetry factor for standard USPLC is not symmetric, so it's changing, and also it's relatively high due to the tailing and a higher error in the analysis. On the other hand, for Nexera XS Inert, you see it's relatively low and also constant, so the peaks were very symmetric. Now in the right hand side is the area over number of injections. For standard USPLC is orange color, for Nexera XS Inert is blue color. You see the area for Nexera XS Inert is relatively high and is constant. On the other hand, for a standard USPLC, it's accumulating the material in the peak, or it's not clearing enough due to the metal. So it's initially low and then it increasing, and finally it's not constant at all. So it will have detrimental effect for the separation as well as quantification. So you can see here the superior performance over standard USPLC for the Nexera access inert. So on the left side, you can see the calibration curve for ATP using a standard USPLC system. And the concentration of the solutions were 1 to 50 microgram per ml. X-axis is the concentration and y-axis is the area. And you can see the RS square value is 0.9918. On the other hand, same sample solutions of the same concentration were used for making the calibration curve using Nexera access inert, as mentioned in the right side. And the x-axis is same as the concentration, microgram per ml, and y-axis is the area. You can see the RS square value is 0.999. So 4 nines, which is almost 1. So it has proven that Nexera access inert shows the quantitative analysis way better than the standard USPLC system. Here is the analysis of oligonucleotides mix containing impurities. Left side shows the separation of how reproducible the analysis was, and the right side shows the individual separations, not for multiple injections. So you can see that 16 mer, 17 mer, 18 mer, 19 mer, and 20 mer are nicely separated, and their separation occurred based on different lengths, different phosphate groups, and based on impurities on free protecting groups. So the separation was done really nicely. Here it is shown the effect of pH change in mobile phase. On the left side, you can see the black color is the day of preparation. Red color is the 2 days after preparation using a bottle cap with a filter. And the blue color is the 2 days after preparation using a standard bottle cap. In all three cases, you can see the peaks are nicely separated. Only the retention time slightly shift. On the right side, you can see mobile phase A is the blue color and mobile phase B is the orange color. Using a standard cap, the B slightly changed because it's organic. But on the other side, cap with a filter is very reproducible. So if you are not sure that what to do, so maybe cap with a filter would be recommended. So here it's shown the rate of change is pH after 2 days of mobile phase preparation compared to the day of mobile phase preparations, which didn't change any of the analysis during this time. Here I will be showing you oligonucleotide analysis by LCMS QTOP. Here I would like to show you some key features of the LCMS 9050 QTOP that will assure you why this instrument is ideal for oligonucleotide analysis. First, ion accumulation capability, which is really very sensitive. And then it uses heated ESI probe, which also enhance the sensitivity. And then it uses reference sprayer, which is used for automatic tuning, so it can tune during the analysis. And then it uses UF grading. UF grading is normally used for advanced ion extractions, which further assure high sensitivity and high resolution. And then it also uses newly designed MCP detector, which is really sensitive, has the long life, and can be used dynamic range. Then it uses high speed digitizer, which is 200 Hertz. That means the ion acquisition is super fast. And then it also uses accurate temperature control that assure a stable mass accuracy. And then it uses IREP top, which is basically an ideal reflection technology, which further assure high precision and sensitivity. Now you are convinced that this instrument will be really useful for your oligonucleotide analysis. It also has a software that is called Inside Biologics. I'll mention you in the later again to explain how that software works. Here I want to mention again UF light tube, which shown on the right side on the top. You can see a tube. This is Shimadzu's temperature controlled UF light tube, requires less frequent calibration, enabling you to run more samples. On the other hand, on the left side bottom, shown the light tube temperature control. So this temperature control light tube and electronics deliver less than 1 ppm mass accuracy without frequent recalibration. Here you can see high mass accuracy and stability for 60 hours. On left hand side, upper panel is the positive mode analysis and the lower panel is the negative mode analysis. In the upper panel, on the top peaks is the peak at room temperature, with mass error is still less than 6. But on the other hand, the small peaks, many peaks together, you see in different homology, those assures less than 1 ppm mass error over 60 hours. Same thing for negative mode, that the upper peak is the peak at room temperature, which is still less than 6 ppm mass error, but for the peaks in the vacuum, you can see they assures again less than 1 ppm mass error. Now let me show you some oligonucleotide analysis by LCMS QTOP. Here I would like to show you DNA oligonucleotide ladder analysis. You can see the ladder 10 mer to 60 mer, all the sequences. This can be used to analyze and identify of your unknown peaks for your oligonucleotides. Here I would like to inform you that we have a new software called Inside Biologics for oligonucleotide analysis and identification sequence coverage of oligonucleotides from QTOP data. This software can be used any kind of analysis and use the inbuilt database that is very much useful for any kind of oligonucleotide research. Here you can see a high resolution mass spectrum of 20 base oligonucleotide. This one I'm showing you as a reference for how other base 10 base to 60 base oligonucleotide looks like. And this is the separation you can see from 10 mer to 60 mer, how nicely they separated. So that means Lab Solution Insight Explode software or our latest Inside Biologics can be utilized to identify and quantify the oligonucleotides as well as sequence coverage and identification of unknown peaks. Here is the determination of molecular mass. First is the mass spectra of oligonucleotide therapeutics, and this is the deconvoluted mass spectra, which shown accurate mass spectrometry using the QTOP mass spectrometer LCMS 9030 determine the molecular mass with an error of 0.003 Dalton. Even using LCMS 9050 can go the sensitivity much higher. Here is the quantitation using MRM mode. First is the 2 nanogram per ml, 5 nanogram per ml, 20 nanogram per ml, and 50 nanogram per ml, and 200 nanogram per ml peaks. You can see that these peaks are the retention time are highly reproducible. So that means the quantitation was achieved very nicely. So that is shown here. You can see that after plotting the curve, it's very linear, and R square value is close to 0.999. That means linearity was observed within a range of 1 to 1000 nanogram per ml, and this was highly reproducible. Now I would like to discuss oligonucleotide analysis by LCMS triple quad. Here you can see that LCMS triple quad, we have LCMS 8040 to LCMS 8060 NX. So in this study, we use LCMS 8060, but it is recommended to use the most sensitive one if your impurity concentration is low, which is LCMS 8060 NX. So data shown here gathered with the LCMS 8060, as I mentioned, varieties of detectors and source options to meet the analytical targets. All LCMS units field upgradable to required sensitivity. And here I would like to show you hardware and method recommendations. You see, this is the LCMS 8060 NX, which has really high sensitive LCMS 8060 NX, and it has ion focus, which is electrostatic field focusing, and then high temperature gas dynamics, which use new ESI heater design and gas flow control, and which has ion optics, that means changes to the field geometry on the quadrupole array, which is called UFQ array 2, and the first quadrupole mass analyzer to maximize higher ion beam densities. So this means this instrument is highly effective to do the analysis in a very low concentration level. Let me show you some results of oligonucleotide analysis by LCMS 8060 triple quad. Here is the quantitative analysis of siRNA. We use two molecules. One is AsRNA, another is ssRNA. You can see the both sequences here. And this is the peak, or these are the peaks from AS oligo and SS oligo. The separation is not very clear, so we used also MRM for assurance of those peaks are coming from individual molecules. This is the analytical curves of siRNA of both molecules, that is AS oligo and SS oligo. Here we used, as I mentioned, it's a C mode, and you can see their quantitation is really good, and it's from 1 femtomole to 10 picomole in a very sensitive way. And their R square value in both cases were close to 1. So that means if you want to do siRNA-based oligonucleotide analysis, this method would be very useful to do the analysis in very low concentration level. Then what are the take-home messages from the webinar today? First, BioInert. BioInert is Shimadzu's latest UHPLC technology, which is the development for the analysis of biologics without compromising any sensitivity that may occur in conventional UHPLC system. This system utilizes metal-free flow path for accurate and reproducible analysis without passivation. IX separation with pH control and monitoring that can be used to assure the reproducibility and error correction if any problem occurs during the experiment. Then LCMS QTOP. For LCMS QTOP, we have a latest software called Inside Biologics for oligonucleotide and its impurity analysis. This software can be used to identify, quantify the peaks, and also do the sequence coverage. This software will be very useful to oligonucleotide therapeutics development and quality control. The LCMS QTOP is highly sensitive and wide dynamic range, stable mass accuracy, and high resolutions further assures oligonucleotide analysis accurately during the development phase. The instrument was applied for the accurate molecular weight determination with the linearity range of 1 to 1000 nanogram per ml. This also accurately separated and identified oligonucleotide with less than 0.003 Dalton mass error. Finally, the LCMS triple quad. This instrument assures high scan speed and UFC per collision for the true UHPLC compatibility. High sensitive and quantitative analysis of phosphorothioate and siRNA were performed using TIM and MRM modes using this triple quad instrument very effectively. Once again, thank you all for attending and participating. We'll send you an email with a link to view a recorded version of this presentation anytime. If you have not had a chance, please fill out the survey questions on the left-hand side to provide feedback and request additional information. Again, we encourage you and your colleagues to attend our upcoming webinars. Have a great day. Thank you.